Welcome everyone. My name is Tulshi Patel and today I have the pleasure of introducing Revity's Cho Source Expression Platform. The Cho Source Expression Platform sits at the core of Revity's bioproduction offering. The platform includes two suspension-adapted CHO-1 knockout cell lines, which are both supported with a full documentation of the cell line history, as well as a comprehensive booklet with all of the protocols and SOPs required for cell line development. The platform also contains our new transposase-based expression system, which is covered in a royalty-free license between our Cho Source cell lines and the Cho Source TNT. Firstly, I would like to talk to you about our founding host cell line. The Cho Source GS knockout cell line is a suspension-adapted CHO-1 cell line where we used our proprietary recombinant adeno-associated virus technology to knock out the glutamine synthetase gene. This host cell line has been established for cGMP manufacturing and has proven performance in expressing many different types of biopharmaceuticals. The cell line can be successfully used in a variety of different process conditions and with different cell culture media, with no constraints. The second cell line in the Cho Source expression platform is the Cho Source ADCC+ cell line. This cell line was generated by performing gene knockout of a fucosal transferase gene in the GS knockout cell line. By knocking out the fucosal transferase enzyme, we are eliminating the cell's ability to add fucosugars to the proteins that it manufactures. Zero fucosylation on therapeutic antibodies and Fc fusion molecules is known to increase the therapeutic efficacy and potency by enhancing the antibody-dependent cellular cytotoxicity activity. The final and latest member of the Cho Source family is the Cho Source TNT transposon technology. This is a two-component system consisting of a transposon DNA vector and a transposase messenger RNA. This expression system offers a more stable and efficient method for transgene integration compared to more traditional methods such as random integration vectors. Cho Source has a very well-respected presence in the biotechnology and bioproduction field. The platform is recognized globally, and we now have customers licensing the products of the platform in all major regions of the world, including Europe, Americas, and Asia Pacific. Our global licensees now stands at over 100, and this is coupled to more than 60 regulatory filing submissions. 8 of the top 20 pharma companies are using Revity's Cho Source platform, and 3 of the top 10 pharma companies are direct licensees of the platform. The platform has been used to bring forward different types of therapeutics. Some of those are listed here on the left. These include antibodies, which range from standard IgG to more difficult IgM, and also includes more novel and complex formats such as bispecific molecules and Fc fusion proteins. All of these therapeutic types can be applied to a number of different disease areas too. These are listed on the right. These range from oncology to cardiovascular disease to autoimmune disease. And finally, there are many successful applications of the Cho Source expression platform, ranging from human to animal therapeutics, and also in the diagnostics and services sector. The Cho Source expression platform can offer you one integrated solution, all from one company. The platform, which includes 2 unique CHO host cell lines and a transposase-based expression system, has proven performance in a variety of industrially relevant settings, and all can be provided in one combined license, which is free of royalties. The platform has a track record with over 60 regulatory filings and over 100 licensees worldwide. And finally, use of the platform is supported by comprehensive protocols and SOPs, as well as a global technical support team. Thank you for your attention. If you would like to learn more about the platform, please contact our dedicated global technical support team at Revity at bioproduction@horizondiscovery.com. I will now hand you over to Sanyou Bio, who will speak about the great work conducted using the Cho Source GS knockout cell line. Thank you.
Hi, welcome to our informative webinar on the development of high-expression stable production cell line. We will cover the best practices, and from our own professional journey, we will share with you our very own case studies. We will cover the key aspects of cell line construction and process development. The central principle in cell line development is to have an excellent host cell line that has the following key characteristics, namely high titer, clear traceability, robust process, feasibility for scale-up, top stability, and quality assurance. All 6 characteristics are important requirements since chemistry manufacturing control, CMC, also known as pharmaceutical quality, needs to be closely connected and monitored. In the next part, we divided the cell line and process development into 3 major stages, including molecular design, cell line construction, and process optimization. The first stage is molecular design, which includes vector selection, codon optimization, and format design. The goal of molecular design is to improve product quality and yield, reduce costs, shorten development cycles, and improve product competitiveness. The selection of expression vectors is crucial in molecular design. Currently, conventional vectors are mainly divided into 2 categories, including screening by the metabolic stress and by antibiotic resistance, of which the screening by metabolic stress using the glutamine synthase GS system has gradually become the mainstream application. There are also some manufacturers who uses the DHFR system and some other antibiotic systems. At present, there are also some novel systems applied to cell line construction, such as transposase system, which include 3 major vendors and products, namely Revvity's TNT system, Lonza's PB system, and Atom's lead-in system. The characteristic GSV0 vector system, independently developed by Sanyou, can be applied to GS knockdown, CHO-K1, naive CHO-K1, and CHO-S, which perfectly matches the screening process of each host cell line. Codon optimization is crucial for molecular design, with rationales including different codon frequency for each amino acid in different hosts, GC ratio adjustment, avoiding enzyme digestion sites, and introducing stable secondary structure to the 5' end of messenger RNA. The general practice is to use online software for codon optimization that is in accordance with the species of the host cell line. Sanyou has a rich experience in codon optimization of both monoclonal and bispecific antibodies. The first case shows that after the early sequence design, codon optimization, and screening method optimization for a monoclonal antibody, the final pool titer increased by 10.84-fold, and transient expression titer was increased by 4.35-fold. The second case shows that though the optimization process for a bispecific antibody, the pool titer increased by 2.32-fold. The screening cycle was shortened by 15 days, and the titer of monoclonal cell line was as high as 7 grams per liter. Isotype selection in molecular design is also very important. The structural and functional characteristics of different isotypes makes a big difference in their therapeutic applications and production processes. At present, more than 75% of listed monoclonal antibody drugs are of the IgG1 isotype, while IgG2 and IgG4 take a smaller proportion. Sanyou has an extensive experience in developing various antibody isotypes, of which IgG1 and IgG4 are the majority. Format design of bispecific antibody vary in complexity. For instance, a bispecific antibody of the conventional format on the left yields 9 types of byproducts, while NOPs into host format might generate 3 types of byproducts. And if one adopts the CH1-CL format of cross-map, byproducts can be circumvented. Therefore, for the same bispecific antibody, different format designs will lead to different byproducts, and byproducts can be avoided by the format design. which makes downstream development a lot easier. In the following case, this bispecific antibody has 5 candidate monoclonal antibodies for one arm and 2 candidate monoclonal antibodies for the other arm. When formatting the bispecific antibody, purity tests show double peaks for any bispecific antibody that contains clone 10E8. Further testing of the individual monoclonal antibodies revealed that 10E8 monoclonal antibody exhibited double peaks. When clone 10E8 was further engineered to resolve the purity issue, we successfully engineered a biospecific antibody of 10E8 that shows single peak when paired with P140. Therefore, if the monoclonal antibody used to format biospecific antibody has poor purity and must be selected, it should first be engineered to meet the quality requirements. Sanyou provides a series of solutions for sequence design, cell line construction, and culture and purification process development for biospecific antibodies of complex formats using the Cho Source host cell line. For example, this target bispecific antibody of the NOP into whole, an asymmetric conformation with one arm as the single-chain variable fragments, would yield 4 types of major byproducts. Firstly, we optimize the plasmid ratio for this antibody by testing the purity of different plasmid ratio for the 3 chains in transient transfection and selected the ratio with highest purity in the main peak. Secondly, during the cell line construction and clone screening, we introduced a screening method to have protein A initial screening and protein L secondary screening, which ensures the expression level but not depleting part of the byproducts. In this case, after one-step affinity purification with protein A, some byproducts and half antibodies were reserved, and consequently, kappa select method was used to remove homodimeric byproducts. Finally, the purity CE main peak was improved from 68% to 87%, and the SEC reached 97%, and there is still potential for further downstream optimization. The second part, cell line construction, mainly covers the selection of whole cell line, screening methods, transfection methods, regulatory requirements for monoclonal origin, and cell line stability. First of all, the selection of whole cell lines. In the industry, there is about 70% of the antibody drugs which are produced using the CHO cells as whole cell line. The history of CHO, Chinese hamster ovary cell lines, can be traced back to 1957, which was divided into 2 categories later, CHO K1 and CHO S. Based on the genetic modification, current mainstream CHO cell derivatives were divided into 3 categories: wild type, DHFR knockdown, and GS knockdown. Currently, bulk pool and mini pool are commonly used in the market for cell pool construction. Bulk pool is to screen a large number of cell clones characterized by short cycle time and high diversity, but with low expression, while mini pool is to screen a small number of transfected cells characterized by generally high expression and long cycle time. Based on the bulk pool and mini pool methods, Sanyou has developed a fast pool screening method, which is equivalent to bulk pool in terms of cycle time and mini pools in terms of expression levels. Next, we will present detailed cases. The first case, when we compared the screening methods, the optimized screening method shortened the cycle from 10 weeks to 6 to 7 weeks. The mini pool screening method generally takes about 10 weeks from transfection to the production of 15-liter samples to get more than 20 grams of products, while the whole cycle of bulk pool takes 6 weeks, and the whole cycle of fast pool is in the range of 6 to 7 weeks. Next, we compared the expression level titles between fast pool and bulk pool. The fast pool screening method showed 2 to 3 times higher expression levels than bulk pool method. Sanyou will choose the most suitable screening method that suits the characteristics of different antibodies and can provide flexible services with the fast pool cell line screening method, especially for complex and challenging monoclonal antibodies, bispecific antibodies, fusion proteins, and other modalities. Sanyou offers mini pool yield with bulk pool turnover. Transfection is the source and the most important part of the cell line construction process. Mammalian cell transfection consists of transient and stable transfection, and the most used method are electroporation and cationic liposome. The selection of transfection method is based on the equipment, cell type, and other factors to achieve the best transfection efficiency and cell viability. The transfection methods that Sanyou is equipped are physical, chemical, and biological methods, all of which have been well-established systems. Regulations in the cell line construction address the requirement for monoclonal origin. Due to the widespread genetic mutations in CHO cells during production, it is critical to minimize heterogeneity in the cell bank, maintain consistency and predictability in the production process, and ensure that the yield and the quality of the final product are within design ranges. The ICH guidelines require that the cellular matrix used to produce an antibody must be cloned from a single cell progenitor. This is a 2-round and the 1-round limited dilution method with plate imaging. The 2-round limited dilution method is simple but time-consuming, and this has gradually phased out, whereas the 1-round limited dilution method with plate imaging does not guarantee the monoclonal rate. Currently, the mainstream practice is to use single-cell plating and well-plate imaging method. Sanyou is equipped with 2 sets of plating and imaging equipment, which greatly improves the monoclonal rate of plating, and the monoclonal rate is more than 85%, providing evidence of compliance for monoclonal traceability. The last key control point in cell line construction is cell line stability. Cho Source cell line can be passaged to more than 60 PDL without selection stress, and the deviation of gene copy number and titer can be controlled under 20%, which is far lower than the industry standard of 30%, and it can support the large-scale commercial production of thousands of liters. The third part is the optimization of cell culture process, which is divided into titer and product quality, control of acid peak, control of aggregates, and semi-antibodies and perfusion processes. The purpose of cellular process development is to ensure process stability and scalability, reduce production costs, and optimize product quality. The process development department at Sanyou can use different optimization strategies like plate bioreactor, shake flask, and 3 to 15 liter bioreactor stages to ensure one can meet the final deliverables. For titer and product quality improvement, we use Cho Source host cells to construct a monoclonal antibody-expressing cell line, and the titer was 6.4 grams per liter, and the CE purity was 87.2% when using the platform process. When we optimized the basal media and feed media, the titer was increased to 8.3 grams per liter with an increased CE purity of 94.1%. Progressively, we optimized both the bioreactor process and feed strategy to obtain a final titer of 13.1 grams per liter and a CE purity of 95.4%. So this slide here shows the detailed data. 0 represents the results before optimization, and 1 and 2 are the 2 different optimization strategies, respectively. The figure suggests that the cell density, viability, metabolites, and the other indexes are kept in a good range, while titer and purity significantly improved. Next, we will look at the regulation of acid peak, which is a challenging issue in CMC development. The causes for acid peak formation is due to silylation, deamidation, fragmentation, glycosylation. The acid peak is usually optimized by pH adjustment, addition of tyrosine, cysteine, temperature adjustment, and other applicable methods. In a bispecific antibody product, we found that the ratio of acid peak in the original process was 37.2%, and the goal was to reduce the ratio of acid peak to less than 30%. There was no improvement in the acid peak ratio through conventional pH adjustment and temperature change. We innovatively used the gradient cooling method from 36.8 degrees to 32 degrees and from 32 degrees to 30 degrees to reduce the acid peak ratio to 27%, and this achieved the development goal. Next, we look at the regulation of aggregates and half antibodies. Cho cell line expressing a bispecific antibody yielded a high half antibody content of 16% during the fed batch culture. To solve the problem of high half antibody ratio, the culture temperature was regulated, and the half antibody content of this bispecific antibody was reduced from 16% to 1% when the culture temperature was lowered from 36 degrees to 32.5 degrees or 31.5 degrees. Although we could control half antibody impurity by cooling the culture, as shown in Figure A, the decrease of the culture temperature increased the aggregates, as shown in Figure B. We further investigated the aggregates' content, the time point of cooling, and the extent of temperature change. The study revealed that when the lower temperature was delayed, the content of aggregates was decreased when comparing the cooling on day 4 to day 5 and 6. When comparing cooling down to 29 and 31 degrees, lower aggregate content was observed at 31 degrees. The final process is balancing half antibody and aggregates by cooling to 35 degrees on day 5. When comparing the culture patterns, biphasic mode, which uses 36 degrees for the initial phase, then cools down to 32.5 degrees, and constant, which is 36 degrees, the biphasic mode had lower half antibody content and low molecular weight content and had higher yields from the cation exchange chromatography. And finally, the perfusion process is a suitable alternative for biospecific antibody projects when traditional fed batch culture process falls short of achieving project goals. The ratio of aggregates, half antibodies, and acid peaks of 6 biospecific antibodies of different formats were examined and compared in both processes. The study revealed that the antibody concentration in the perfusion culture was lower than the fed batch culture, which may also be the reason why perfusion can reduce the proportion of aggregates. When we examined the half antibody and acid peaks in perfusion culture, the antibody molecules were in the culture medium for a shorter period, which may be the reason why perfusion can reduce the ratio of half antibody and acid peaks. Perfusion process can effectively regulate aggregates, half antibody, and acid peak content for most bispecific antibodies. But there are some bispecific antibodies that will have better results in fed batch culture, which requires further consideration according to the antibody properties. This slide examines how perfusion culture enhances the quality of bispecific antibodies. Compared to fed batch, perfusion culture reduces the intracellular accumulation of reactive oxygen species and the oxidation of glutathione oxidation. This suggests that the maintenance of glutathione levels and intracellular regulation can protect the CHO cells from the pressure of the endoplasmic reticulum stress response, which eventually reduces the antibody aggregate content. In summary, here are some key points for improving the purity of the bispecific antibody. The format design. An optimal format can improve the quality of bispecific antibodies. Gradient lower culture temperature can regulate the content of half antibody in bispecific antibody. Perfusion culture process can improve the yield and quality of bispecific antibody. When the purity of the two monoclonal antibodies does not qualify, optimizing modification of the monoclonal antibody shall be considered before forming the bispecific antibody. Metal ions, amino acids can affect the quality characteristics of the bispecific antibodies. And finally, the reductive breakage of disulfide bonds can be improved by perfusion and additives during the supernatant harvest phase of the cultures. Now, we would like to introduce Sanyou's all-inclusive preclinical candidate, PCC, to investigational new drug IND service. We can integrate every stage from PCC to IND, offering comprehensive R&D support for innovative antibody drugs. You can either opt for the whole package or the stage service. Our experienced team has promoted several drugs to market, managed over 30 ongoing PCC to IND projects, led or assisted 6 projects that completed IND filing. Notably, we also achieved US-China pre-IND filing for an anti-COVID-19 bispecific antibody project within 12 months. Our core advantages are 4 sets of authorized GMP cell lines for production, innovative fast pool screening method, protein expression exceeding 13 grams per liter, covers a variety of innovative drug modalities, and customizable services. Our service features include cell line construction, process development, which is upstream and downstream, purification, formulation, pilot production, toxicology studies, validation, and verification. All the steps are meticulously coordinated for 12 to 15 months delivery timeline. This is an image of a production cell line construction laboratory, where cell line construction is performed in a C+A grade environment. We also have 18 bioreactors for clone screening which allow annual throughput of up to 30 projects. Please feel free to contact us if you have any inquiries or interests for collaboration. Thank you for your time and attention.